now we'll go for the next group of sugars and carbohydrates. We'll test it. And under this, we are going to test reducing sugar. Till now, in the last class, we have seen non-reducing sugars. Now we'll go for the reducing sugar. Reducing sugar, example, we'll test for glucose. And for this, we need to have two important tests. Number one is Fehling's test and another one is Benedict's test. For Fehling's test, as usual, we will take three different test tubes and three different samples will be given. And in each test tube, we will be adding 1 ml of the test solution. Okay. And after adding 1 ml of test solution in each test tube, then we will add 5 ml of Fehling's solution. Fehling's solution is a ready-made available commercial product and that will be added to the sample. Once the Fehling's solution is added, we will boil the sample for few minutes. After few minutes, there will be a change in the color of the sample. And generally, the sugars will give rise to a brick red color. Reducing sugar will give brick red color, indicating the presence of the glucose. Similarly, we have one more test that is called the Benedict's test. For Benedict's test also, we will be taking as usual 3 different test tubes and 3 different samples will be taken. And in each test tube, nearly 1 ml of the glucose solution. Okay, that is unknown sample as well as the known sample will be taken. And after taking the samples into 3 different test tubes, 5 ml of Benedict's reagent will be added. Mix well the sample and boil the sample for few minutes. Few minutes present, we are going to do it for 5 minutes so that we can observe the change in the color of the sample, either orange or red. Depending on the amount of glucose present in it, the color will vary. If less amount of sugar is there, orange color will be given, and if more amount of glucose is present, then it indicates the large amount of glucose. Okay. So it is of reducing sugar practical. Right. Now we go for the demonstration of the same experiment as usual. Now we will go for the demonstration of reducing sugar analysis. First test is Fehling's solution and second test will be Benedict's solution. As usual, Mr. Varun will show you how to take the samples. He has taken 3 different test tubes and labelled it as ABC and 3 different samples ABC, and also the Fehling's reagent, commercially available Fehling's reagent, and this Fehling's reagent he will be using for the analysis of the experiment. Okay. Now he is taking sample A into test tube A. Sample B in tube B. Sample C. Sample C in tube C. Then he will add 5 ml of Fehling's reagent in 5 ml of Fehling's reagent in each test tube. Then he will go for the heating of the sample. Now as usual, the samples are heated in the hot water bath and we will wait for few minutes so that there will be a change in the color to brick red. Okay. After 5 minutes, there is a change in the color of the sample. Okay, we'll come in the light so that we can observe. For the first sample, that is, he got the brick red color, and the other 2 samples he could not get any color. That indicates the wherever you found the brick red color, that indicates the presence of reducing sugars. Test done by Fehling's solution. Now we go for the second type of test for glucose or reducing sugar. As usual, Mr. Varun, he will take 3 different test tubes and 3 different samples, and in each test tube he will be adding 1 ml of test sample, that is glucose, and then he is now taking the samples in each test tube. Sample A in sample test tube A, sample B in test tube B, and a sample C in test tube C. After adding the samples, he will be adding Benedict's reagent, 5 ml of Benedict's reagent in each test tube. Benedict's reagent is commercially available. And in all test tubes, he has added Benedict's reagent. Okay, that is 1 ml of the sample and 5 ml of the Benedict's reagent. Now he will go for heating of the sample for few minutes. As usual, Mr. Varun is placing the test tubes in the hot water bath for boiling for few minutes. After boiling the samples, wherever the glucose molecules are present, it has changed to orange color, but when there is no glucose, the Benedict's reagent remained as it is. There is no change in the Benedict's reagent. But when the glucose is present, it has changed to orange color. That is the confirmatory test for the reducing sugars. That ends your carbohydrate analysis. Thank you.